Kindly mute your audio and video. Dear participant, once you join, kindly check that you are muted your audio and video. Yuvrani Vegetation, ma'am, kindly stop presenting. Rejoin. Rejoin yourself. Yuvrani Vegetation. Can you able to hear me? Yuvrani Vegetation, ma'am. You are in the presentation mode. Kindly rejoin. Click join. Not in the presentation. Yuvrani Vegetation. Yuvrani Vegetation, stop presenting. Yuvrani Vegetation, kindly stop presenting. You are in the presentation mode now. Can you able to hear me, Yuvrani Vegetation? Still you are in the presentation mode. I request the participant to pin the today's resource person, Mr. Kalidasan Vasudevan sir. You can find him in the people list. Sir will be in the below. Thank you, Yuvrani ma'am.
Sarvan sir, start பண்ணலாம்.
Yes, father. Ma'am, Shamla ma'am. Kalidasan sir.
Yeah, I'm online.
Yes, sir. Yes, sir. Ma'am, Shamla ma'am.
Shyamla ma'am.
Ma'am, Shamla ma'am.
Shyamla ma'am. Ma'am இல்லையா?
One minute, sir. Ma'am.
Hello. Yes.
Yes, ma'am. Yes, ma'am. Yes, ma'am.
Yeah. Okay.
Ma'am, shall we start, ma'am? Start the
Yeah.
program.
Sure.
Yes, yes, ma'am. Yes, it's time to start our program. We, PG Department of Commerce, thankful to our founder, Reverend Father Dr. G. Arulraj, management, our administrator, Reverend Sister Dr. K. Santigamary, Principal Dr. R. Ruben for motivating us, Principal Mrs. Shamla Alexander, Head of the Department, Reverend Father Dr. Adiklasamy, colleagues, participants, and my dear students. It's time to start the International Webinar Series 3 on the topic Trade Distinguished in Global Disaster. I would like to explain why we framed this topic for the entire webinar series. Any field of study need commerce. It is the distinctness of the commerce department. With this honor, I invite our head of the department, Reverend Father Dr. Adiklasamy, to lead us in prayer.
Thank you, sir. Let us bow our head and put ourselves in the holiness of God, the Father, the Son, and the Holy Spirit. Dear Almighty and ever-living God, we glorify and thank Thee, Your name. You have showered us with so much blessings, and Your presence continuously remind us of Your faithfulness and guidance. We humbly ask You to shower our speaker, Mr. Kalidasan Vasudevan, today of Your greatest inspiration, so that he may share the most of his knowledge, heart and soul to their respective topics. May we also absorb the invaluable knowledge, experiences, and put it into practices what we may learn today through our international webinar series. We pray that You bless all the committees in charge, especially PG Department of Commerce, staff and students, that they may be able to fulfill their task responsibly, that the objectives they have set may all be achieved. Your infinitive blessing would mean the success of this international webinar series. May we be a living witnesses of Your genuine love through the enactment of the knowledge acquired through this international webinar series. We ask this in the name of Jesus, our Lord and Savior. Amen.
Yes, thank you, Father. Personal and career development always grow when we update our knowledge. The person who taught us, Mrs. Shamla Alexander, our Vice Principal of the college, will deliver the welcome address now. Ma'am, please, ma'am.
Good morning to one and all. A small chief welcome makes a big face. Distinguished chief guests, secretaries, staff members, and participants, I am sure that today to be a memorable day for each one of us. On behalf of St. Joseph's College, the PG Department of Commerce feels proud in welcoming Reverend Sister Dr. K. Sadhya, Principal Dr. R. Ruben, Vice Principal Ms. B. Vijayalakshmi, and the Head of the Department of Commerce to this session with the topic Harnessing Gene Editing for COVID-19. It's my pleasure to welcome our eminent speaker of the day, Mr. Kalidasan Vasudevan, University State, Malaysia. I cannot think of another person more qualified than him to be our resource person today. I extend a warm welcome on behalf of PG Department of Commerce, sir. Let me raise up a hearty welcome to all the heads of various departments, staff members, and participants. We are so excited to have all to this webinar. I welcome you one and all. I wish you all a pleasant and fruitful day.
Thank you, ma'am. Thank you so much, ma'am. I request the participant before getting into the session to mute your audio and video. I can see Miss or Mrs. Kazila Farah, ma'am. You are in the presentation mode. Kindly go and rejoin yourself. Kazila, ma'am. Kazila Sarah, ma'am, can you hear us? Kindly rejoin. I request the participant to mute your audio and video. Yes, it's time to hand over the session to our resource person, Mr. Kalidasan Vasudevan, sir. Sir, can I hear me? Can you hear me, sir?
Yes, exactly. I can hear you.
Yes, sir. Please, sir, take over the session.
Okay, thank you so much. Thank you for the wonderful experience for this day, and thank you for everyone that is being in part of this webinar series. And I appreciate for wonderful opportunity given for me to share my knowledge and experiences on the topic that I will be presenting today. And to start with, I'll just give you a brief introduction about myself. I'm Karidasan Vasudevan, and I'm doing my PhD right now at the University of Science Malaysia. And I'm done with my MSc in medical microbiology and Malaysia itself. So currently, I'm focusing my studies on HIV and also something called um, gene editing technique. Today, my presentation will cover how we can use gene editing in regards of the COVID-19 pandemic disease. Since this topic, it's more of like more life sciences, and I will not go into too much of the complicated version of the series because I believe that there's a range of participants starting from arts and sciences. So I'll cover as brief and fundamental as I possible. Okay, so before that, I will start that. Okay, so once again, I'm Kalidasan Vasudevan, and I'm doing my PhD, and I'm from Advanced Medical and Dental Institute, University of Science, Malaysia. This university is located in the northern of Malaysia, which is, if you know where is it, it's located in the Penang region. So today's topic is on harnessing gene editing, and particularly we are talking on the COVID-19 pandemic that is affecting many countries around the globe, and we are actually going through this phase of all the containment, all the treatment, and also prevention right now. This presentation will cover the basic of what basic of biology is all about, something particularly looking at the basic of molecular biology. So I'll touch a basic of molecular biology, that it's important to know that before we can understand what is the next second portion of the presentation, which is on the gene editing. In simple term, gene is some part of the DNA that we have, and editing is basically we want to make changes to that DNA. And on the third portion of the presentation is basically covering on the pandemic of COVID-19. Even though we know most of what is COVID-19, what the virus and symptoms, and what we can do about it, I'll recover again, just reminding all of us that this is what coronavirus disease and the all the disease process look like. And finally, the important is relating the fourth portion about gene editing, that we look at the second part with the third part, and how gene editing come in place in terms of diagnostic of laboratory diagnosis particularly, and how gene editing can be used in a vaccine development. So before that, let me start on the first part of what is the basic molecular biology is all about. Okay, the concept of molecular biology is important if we all learn about biology, and particularly looking at molecular refers to the very basic or fundamental of what we are, the humans, the organisms, or the living plants and animals all around us. So in a very simple term, molecular biology refers to the DNA, and DNA is the molecule of life, because without DNA we are not here right now, the animals around us are not here, and the plants as well. So as we, the human example, we are the organism. A simple term is a form of life. A human is a form of life. Plants and animals are also a form of life. They can do all the living processes, like we can breathe, we can eat, we can digest, we can actually respire, we can breathe, and we can reproduce for the next generation. So this is basically what organisms should do, because they are a living organism. Zooming into an organism, for example, human like us, we are actually made up of cells, and cells are said to be the basic unit of life, the fundamental unit. Without cells, we are not a living organism. So example you're seeing right now, it's a cell. and we can see in the middle part, which is called the nucleus, which have the genetic material. So genetic material, as simple as right now, we can see something called chromosomes. So if we zoom into the nucleus of a cell, we will know that we have the chromosome, and chromosome are basically they are packed together. They are packed because all the DNAs are coiled. They are packed together to form a very tight complex, which is called the chromosomes. And this chromosome is located in the nucleus of a cell, and the cell is located in the organism. If we can open this chromosome, we can basically unveil the important molecule of life, which is the DNA. And DNA is basically appear to be in a coil structure, which is called the double helix. And DNA is the genetic information that all of us, the organism, be it animal, plant, or human, must have, because that carry the genetic information that can be passed from one organism to another organism to the next generation. And this determines how we are, how the characteristic of our physical characteristic that we have. So moving on the concept of molecular biology, I'll talk a little bit on the human genome. So what is basically genome is, as defined, is genome is the whole of its hereditary information encoded in its DNA. So we collect all the DNA. Basically, we can see that the DNA collections are called the genome. And genome is very important because that's determine the healthy level of us, the disease causing every single thing that happen in the body is because of the genome that we have us. And genome also being affected by the lifestyle that we have, the food we eat, the type of work that we do, the stress level that we have, and a lot of things that happen in the biological system. If we're given a chance, like we can cut our DNA into small fragments, and we can actually label them according to some color coding procedures, and through some instrumentation we can actually analyze all these fragments, all these genes, and particularly we can learn a lot of things through this human genome. And this come to an idea about the concept of human genome, where Human Genome Project was done to determine the DNA sequence. The sequence is referred to the DNA fragment that human have, and basically it will tell you the gene that is related to cancer, related to some diabetes, related to certain illnesses in our body. And by identifying this gene, we can actually do a lot of marvelous thing about treatment, curing, and so on and so forth. So that's what Human Genome Project was done in the past, to know what the gene is in human are doing. In medical application, and what is the benefit that we can get is we can actually improve the diagnosis of disease. Since we know that disease is part of human life, and we can improve, we know certain disease have certain genetic predisposition. So if we know that genetic cause of the certain disease, we can actually do something about it, so we can improve the diagnosis of disease in a very quick manner. As I told you, in a quick manner, we can do some detection, early detection of disease, so that the disease can be treated in early. For example, cancer in particular, where some patient we have cancers causing disease, cancer causing genes that will be detected later, and they will actually resulting in death. So if we could detect these cancer causing genetic material, early diagnosis, early treatment, and saving life is possible. Create drugs based on the molecular information is also something that is coming important, where the molecular information, like the DNA, will tell you that this drug is suitable to be used because they can have a good therapeutic level, so that the particular drug can be given to the patient. These come to rise to a design of custom drug. So based on every genetic individual, we all know that each of us are having different genetic information. Like person like me and another person is quite different in certain genetic information. So certain treatment that given to me, it's not considered good for another person. So we don't want to give a very standardized treatment to everyone. For example, for a certain cancer treatment. So we come up with a certain customized, which they personalized for each person based on the genetic information. Genetic information will be revealed, and we study the genetic information, and based on that genetic information, this person will be given a custom drug, and that drug will basically gonna play an important role for the treatment. And the last part of why we need this human genome is for particularly about the treatment. There's nothing much where it comes to treatment in sense of where gene therapy is coming in. So which will talk further on the gene therapy and how the human genome is helping for treatment. Coming back to the molecular biology, we have something called central dogma, the central of molecular biology. We basically that all the information are being transferred from one level to another level. How like a DNA information can be transferred out until the human is look in this way. Our eyes is in this color, our hair is in this color, the skin color is in this tone, our hair color, all determined by all this DNA. And how DNA is giving this information is all about this central dogma. So it's basically a flow of genetic information, as I told you, from one level to another level. And this will be started by the DNA, the genetic information, will be the information will be then transferred to intermediate level, which is called the RNA. And basically RNA is said to be the expression of gene. If DNA will give the code, and then the RNA will actually express the gene in sense of the RNA. And finally, RNA will be actually flow with the genetic information into the protein, which is the functional product of everything that we have around us, be it all the food digestion enzymes, be it all the skin material, the hair material, all basically the proteins. And also the important right now is the antibody is also the protein that actually fight against the infection. So we can see that the genetic information is flowing from DNA to RNA and to protein. And basically there are few steps in between all of these. And we see the first steps, which is called the transcription, basically copying a gene DNA. We copy the DNA and we transfer it to an RNA. And this process is called transcription. So basically decoding back the information in a form of RNA. And RNA will be converted or the information flow into a protein by a process called translation. Translation is basically making a protein and using an RNA as a code or template for the genetic information. So overall DNA to RNA by process of transcription, RNA to protein by process of translation. So this is what a basic central of molecular biology looks like. Next we come to another technology, or we can say that another component of the biotechnology is basically omics technology. So omics technology is basically a technology that can use to measure the characteristic of cellular molecules. So cellular molecules that I'm referring here is DNA, RNA, proteins, and a lot of other molecules that present in the human body. And we can actually analyze them, we can study them, and we can understand further what their function is, what's the probability of involvement in the human system. So coming back the omics technology, we can start always with the DNA, which is genetic information. The omics technology of DNA is called the genomics. And the next level, which is RNA, the expression of gene, the omics technology is called as transcriptomics. And the third level that we know is protein, which is the functional product, and the omics technology is called proteomics. And the last is called the metabolites. So metabolites is basically the end product of metabolism. Example is we are having our food daily in sense of carbohydrate, protein, fish, rice, noodles, and bread that we eat every day. Basically will be digested into small product. And that small product will basically called the metabolites. And the study of metabolites in sense of omics technology is called metabolomics. So these are the technology and the each level how the measurement can be done in certain of RNA, DNA, protein, and metabolites. Coming back to why we need to study all this omics technology, why is it important to study DNA, RNA, protein, and metabolites? What it's going to tell us in basic level. So to study all of these and correlating all of these technologies, we say that it's multi-omics integration, where all the omics are integrated together to give you some idea or give you some interpretation of all about human and human processes and also the human disease. So multidimensional understanding of health and disease biology is taking place through the multi-omics integration. So we know that human are made up of living things, cells, as we saw, chromosomes, and all the DNA, RNA, and proteins and metabolites are being part of these. Multi-omics technology that we know through genomics, transcriptomics, proteomics, and metabolomics are basically going to give a lot of idea. So the genotypes, the term here they're referring to is the genetic makeup of a cell. So genotypes refer to the genetic information that the human have. And through the level of this multi-omics technology, it will be visible, it will be interpreted into the phenotype or the physical characteristics. So as I told you, how the DNA in the body will tell you the color of our eyes, the hair, the personal trait of us, the skin color, a lot of physical characteristic that we are seeing right now because of the genetic makeup. If we know the genetic, we can determine our physical characteristics. So using this genetic information, we can also learn about the disease. We can learn about the health biology in the body. So in a way, learning all of this, integrating the multi-omics will give you an important achievement, which is the therapeutic and clinical outcome. So in sense of clinical disease and the treatment required can be actually personalized and can be categorized into a more advanced level if we use all the information through the multi-omics. So in a way, in a very simple term, we can learn about the health, we can learn about the disease when we know the multi-omics information. So the key summary, I know that it's quite detailed and it's quite, even though it's fundamental, it might be confusing because a lot of terminologies are involved. So I just recap the first part of the summary by providing a very simplified version of the presentation of the first part. So we know that the molecular biology relating with DNA, which is the genomics in the omics technology, and the RNA, which is the transcriptomics, protein, we say the proteomics, and metabolites, the metabolomics. So the scientific question that we can know in level of DNA is what can happen? In a very fundamental level, DNA is the basic. So it can tell you, it give you a code what can happen in the body. So it's basically the boss tell you that the disease can happen because there are certain disease causing genetic material. And if that material being passed to the next level, flow to the next information, what appear to be happening is the question here, because that DNA level information is being translated, transcribed into a RNA, and that RNA now hold a code for maybe a disease causing problem. And if that RNA carrying the disease causing question is that the protein will make sure that it will be shown up. So the disease problem is coming into the real situation. You can see the person have symptoms because of the protein expressed in the system. So what make it happen is the protein level we can see. And at the metabolite level, we can say that after cancer, we can see certain cancer will release certain material from the tumor, from the cells, and that cells is basically is the tumor cells. And we can actually detect certain material that being released from the tissue, certain product that being released from the tissue, and it will tell you what has happened. So basically, the body has actually undergone a cancer production, and then the body is basically producing cancer cells, and that through these metabolite that the body is producing, we can actually do diagnosis, certain detection, and also treatment as soon as possible. So we can say in overall, I've given an example of a cancer, that how genetic changes in the DNA will be flow into an RNA flow into a protein, and a protein to a metabolites, and overall tell you what have happened in the body, what can we do next about the disease process. So this is about the summary of the first part of the molecular biology. If we know this part, the next part will be more easier to understand, which is the basic of gene editing. So gene is the particular part of what we saw in the DNA, and editing is basically changes, any changes that we want to do into the DNA level. So gene and function alteration. So if we alter the gene, the function will be changes. If we have gene X, we change that, the function overall will be changed. So it's a process of changing a DNA which result in alteration of a function. So this process, in a very fundamental level, is basically on the DNA, and a person or a technology is being used to change a portion of it. We can actually cut, we can add, we can modify in the way that we want it, and overall, when we do that, the function will be altered. We can change the disease to a healthy, and healthy to a disease, depend on the situation and the clinical diagnosis that is required. So we have seen there are 3 alterations that can be done. First one is called the knockout. So knockout is basically refer to removal of gene. So we are removing the portion of a gene. For example, the example is quite easy, it's about cancer. So cancer, we know that a certain portion that is responding to the genetic response to the cancer progression. So if we know that certain gene is responding to that, we can remove the portion of the gene by process of knockout. So the gene causing material will be not there, the patient will not progress into the disease. In another example is knock-in, we refer to introduction of a new gene. So we know that certain illnesses require certain alteration. We want to enhance the immune system, for example, or in case of HIV. So in HIV, we know that immune response is greatly low and they are quite easily to get be infected by other illnesses. If we know a certain immune protein that is important for the body, we can put in the gene responding for these good immune responses, and the person can actually have a good immune responses. So we can introduce new gene, a good gene, a beneficial gene if it's required. The third example is called knockdown. It's not removing out or putting in a new thing, but it's just basically we just suppressing it, reducing the expression. So we are basically silencing it. It's there. Basically, we just say that you can't do anything much. You have to be silent. So that will be result in no production of any expression. There will be no changes in the human body. So basically, we're just silencing it for a moment, and we can lift that silencing when the time is come, according to requirement. So here we have knockout, removal, knock-in, introduce new gene, and knockout, which is silencing or reducing. Coming back to the gene editing platform that we are looking at here, we have quite a number of platform or techniques which I refer here. The famous gene editing platform I'm referring right now is something called the clustered regularly interspersed short palindromic repeats. Okay, the name is quite mouthful, and I know that. And what does this really mean is quite a complicated term. So in sense, for this level, it's good for us, like a public person who non-biological person, to know this gene editing as CRISPR, CRISPR. So if you know CRISPR is, this is what CRISPR is basically a gene editing technique or a platform. So you can see in a diagram, basically there are something like scissors being shown here, and basically doing some cutting. So in a further explanation, I will tell you what this scissors is doing and what changes are we looking at. Something about the history of gene editing, which is this CRISPR is all about. It was first reported in 1987. The scientists found that CRISPR is actually being present in the biological system, and which biological system I will explain in detail later. In 2002, the scientists gave a name, CRISPR, which is the clustered regularly interspaced short palindromic repeats. As we know that every single thing needs a name, so they named CRISPR for this technology. And in 2013, scientists started to use CRISPR to edit human genome, particularly in animals and plants at this level in 2013. Because of CRISPR is basically changing human genome, there's a lot of ethicals and also the societal application and permission that is required to do experimentation of human. So 2013 is the embarkment of CRISPR technology. So CRISPR gene editing, I'll give you an explanation based on an analogy, so that we know that CRISPR is basically can be understood in a very simplified version of, rather than scientific explanation. So I'll give you an explanation. It's basically CRISPR technology is basically how we use your word processor, your Microsoft Word. It's just placing a cursor between two letters and hitting delete or clicking paste. So we want to basically edit the word, a sentence in your word processor, or we want to include a new term in by pasting method. So it's basically changing in that way. So I'll give you the example by using rhymes. So all of us know these rhymes. When we are when we are small, we learn about this twinkle twinkle little star. But in here, there's a problem with the lyrics given here, which is it's shown that twinkle twinkle big star, and we know that it's supposed to be little star, not big star. So when coming to this example, we wanted to change this lyrics, because we know that twinkle twinkle little star is the correct one and not the big star. So why don't we do some changes? So first method is we can remove the big with the word big. So when we remove the word big, it becomes twinkle, twinkle star. And this is not okay still, because it's supposed to be twinkle, twinkle, little star. Next is, why don't we add little into the original version of twinkle, twinkle, big star? When we add little into the sentence, it's become twinkle, twinkle, big little star. And this is still not okay, because it's not the original lyrics of the song. And when we come to the CRISPR technology, by using CRISPR gene editing, we know that the original was, the error was twinkle, twinkle, big star. And we're going to cut the word big by using CRISPR. So when we cut the word big, it becomes twinkle, twinkle star. And the next thing is, we just need to add the word little. So when we add the word little, it becomes twinkle, twinkle, little star. Then that's fine. That's okay, because the original lyrics is correct. So we can see that CRISPR gene editing works in this way, where you need to cut the error portion of certain sequence or certain letters in this way, and can do some repair. Or in the case we want to add something which is the correct version of certain things, and we can add the correct version, the correct sequence into the genome or the DNA. So CRISPR is basically working as a scissors, cutting it and then put the correct version by repairing it. So this is what CRISPR can do. So as I told you, CRISPR is basically being in the human, not in the human, but in the living system. And particularly CRISPR was located in the bacterial immune system. Like human, we have immune system. Bacteria around us also have their own immune system. And particularly we are looking at the bacteria, which is always can be infected by viruses. So system that the bacteria use to defend against the viral attacks. So like human, like us, we can be attacked by bacteria and virus. Bacteria also have problem because virus can attack these bacteria. So these diagrams show, you can see a virus, which is the green color, and appear to be look like a spider, which is called the bacteriophage. Can infect a bacterial cells, the blue color. So in this process, I'll guide you that according to the numbering. In the first level, you can see that the virus try to invade the bacterial cells. So it will infect the bacterial cells. And the second level, you can see the genetic material is being released into the cells. When the genetic material then it released and then later being integrated into the bacterial genetic material. So the bacteria have their own material, the virus have their own genetic material. These two materials start to fuse together, particularly at the CRISPR location. So in the bacteria, they have something called CRISPR location. At the CRISPR location, the system will later do some modification to come up with a formation of CRISPR RNA. So RNA we know, and CRISPR is the location where the particular system is being located, and they're going to produce this CRISPR RNA. And together with another scissors that you can see in the diagram, basically the RNA, the CRISPR RNA, and together with the Cas9 enzyme, which is a scissors-like, gonna fuse together. They can come to a certain complex, and they can now form a CRISPR-Cas9 complex. And this complex will present in the system of the bacterial cell. And again, if the virus is infecting the bacterial cells once again, in subsequent infection, subsequent infection, the complex will later cut the genetic material by virus. So this process is basically how the bacterial immune system are actually looking at the CRISPR component for it to get evade from the infection again and again. So when the scientists found this system is quite amazing, and they thought, why don't we actually adopt these into a research, into a laboratory base, where we can use this modification or editing of these material to a lot of biological processes. So that's how they come with this CRISPR technology, where the bacteria teach us this technology to human. So coming back to the important part of this CRISPR technology is basically the component, what the CRISPR is basically made up of. So these technologies say to be faster, say to be cheaper and more accurate techniques of editing DNA, even though we have the previous old generation editing technique, but the current one is CRISPR because it's cheaper and faster. So in this case, the component are two. The first one is something called the guide RNA, and a very simple layman term is something called a flag. So flag is basically you can some identification or some recognition. If you see a flag somewhere, then you know that's giving you some recognition. An easy example, if you go for tourists and the flag being carried by the tour guide, you know that this is the mark. Okay, similar, that's the interpretation that I'm using here. So guide RNA is basically a flag that is designed to find a specific place. For example, we know that this gene is causing cancer, illness. We go and find the space, the specific spaces, and we tag them. We put a flag at this particular gene, and then the complex will go and do a lot of other changes. So it will bind to a particular places in the gene, and giving you an idea that this is the changes that need to be taken place. And the next thing is the Cas9 enzyme. So as guide RNA stand for like a flag, Cas9 enzyme gonna act like a scissors. So we have a guide, we have a tour guide tell you this is the place that you need to go. And the next thing is the scissors will go, particularly cut the strand of DNA, and later will be removed out of the region. And we can also add anything that we wanted to add. So the component of CRISPR is basically a guide RNA, which stand for a flag marking of the region, and Cas9 enzyme is like a scissor. We go and cut the particular place, and these two combined together form a complex as simple as called as CRISPR, the Cas9 complex. So how we're going to work in the sense how this cutting and how this guide and marking is being done. So DNA repair going to do all of these processes. Marking, cutting, and repair will take place. It's a normal body processes. When there's some changes happen, the body will do the repair. So first is we need to allocate the specific region, specific disease causing, for an example. So when we know the target sequence location in the DNA, the next thing is we call the guide, which is the flag, go and bind to the particular location. So this is how the binding going to look like. So the guide RNA will bind to the location. After bind to the location, it will call the scissors to come, or the complex to come. So the complex will first bind to the location, and the scissors will do its process, which is cutting. So it will cut at the location that is the guide tell you to cut. When the guide already done with the work and cutting is already been done, repair will be done. So the repair where the cut is there, and then the cut will be covered back, and there will be some changes, and usually a mutation will happen. So mutation can be a good thing, also can be a bad thing. So mutation in the sense of there will be changes in the reading, and if the reading seems to be not a problematic reading and the disease causing can be removed, and that's okay. Some mutation will cause a lot of other problems to the whole system. So we must make sure that the binding of the flag is at the correct place and cutting happen at the correct area, so that the mutation can be reduced as much as possible. So we were talking about this cutting phase, introducing this guide and flag and so on. So what is the basic important? What we can do with this all gene editing? What we can do in the application of this? Basically, it can be applied in many technologies. For example, it can be used in for drug development. Gene surgery can be done. We know that certain gene is causing a genetic problem, changed accordingly, and we can create biofuel from production of any plants and algae, and we can produce food of all these crops that can be against infection. We can produce materials from that is useful from synthetic materials, and we can learn about what causing disease and how we can do some changes. And finally, we can learn about disease, about healthy using animal model. So we can do all of these by understanding gene editing, starting from the food, from gene surgery into the animal model for all the advancement in biotechnology and medicine. So the issue in gene editing, there's always a problematic when it comes to gene editing. So there's always ethical and practical application that we need to consider. So gene editing is something about cells, and there are two cells in the human body that I'm talking here. Somatic cells refer to any other cells, your muscle cells, your skin cells, your blood cells. So this doesn't include your reproductive cells. Germ cells refer to all the sperm cells and the egg cells, all the reproductive cells. So if you want to do changes on the somatic cells, it's just changing a cut and put in, and at this level only the editing will happen at the cell that we are making changes. So in this case, the gene will be not, the editing will be not passed to the next generation. So the next generation is not affected. So in terms of regulation and testing, there will be no much problem. So there will be no much problem on the ethical, on the practical application, because it's just changing the cell of the individual, which is adult cells. So the next generation is not affected. But the germline, we are doing changes at the ovum and the sperm cells of the reproductive cells. So you can see all the cells being changes, being cut, and the copy will be actually passed to every cells in the body. Since this will be passed to the future generation, all the offspring, the daughter and sons of the next generation, there's a problem with the legal and societal issue. People said, how can the next generation being changed just because your parents are being altered? So the next generation should not be altered. There's also religious challenges, there's societal challenges, there's also ethics challenges coming in. So which cells can be edited at the moment? Somatic cells can be edited at the laboratory level. Germline is being a problematic, so researchers are not allowed to do on germline changing the ovum and the sperm cells. So the key summary of the second part is basically learning about how the guide RNA, the flag, the Cas9, which is the enzyme, which is the disease is going to cut, and they can change the gene, or we can include the new gene portion into the DNA. So this is what gene editing is basically changing according to the requirement of what we want to do. So the next part is basically telling about the COVID-19 pandemic. So I will just go through these quite quickly, telling what COVID-19 and biology behind it. So COVID-19 outbreak happened and first was identified in Wuhan city in November 2019 in China. Because of these, a lot of later countries are being affected. And in 11 March 2020, WHO announced that COVID-19 has become the pandemic of the whole country. And as of 2020, 31st May, we can see that about 6 million of cases have been confirmed, and we have about 3,000 cases, and we have about 2 million people have recovered. And coming through the country that being affected, we have about 16 countries exceeding the China cases, because China was the initial places where it started. So we have 16 countries, they are battling through. Even though the countries were exceeding the China, a lot of things have been done to make sure this epidemic is not going into a more worst case scenario. The causative agent that we are looking here is the coronavirus, as we all know. And coronavirus is actually appearing to be like a crown-like. So it's basically like wearing a crown that you can see in the diagram under the microscope. So if we look at the virus itself, the virus is complicated. Like human, we have a lot of portion in our body. The virus also have certain portion in their bodies on their structures. So we have like spikes on their body, which is important for them to fuse with the human cells. So like how a key and a lock, we have a key and we have a lock, similar like the virus is having the key, the lock is on the human cells. So to open up the cells and they can get into the human. We also have component that help it to attach to the human cells. We have the envelope protein for it to maintain the shape, maintain the structure of the virus. And we know that the virus have the genetic material, and the genetic material is an RNA. Like human, we are DNA, the virus have an RNA material. So this is the basic how the virus and how the structure of it look like. So we are looking how the life cycle. So once the virus infecting the human cells, what it's going to do into the human body. So virus enter through the nose, the mouth, and the eyes. So there are the portal of entry into the human body. So the human body have certain complexes inside the human. Process can take place. So when the virus try to sit, it will sit onto the they have the key, which is a spike, and the lock that human have is something called the ACE2 lock. So once it sit, it open the cells. It will try to go into the human cells. So first it enter to the vulnerable cells at the airways and the lung, and try to go into releasing the genetic offspring to produce more of their progeny and basically release more and more virus particle in the body. So this is what happened once a person is infected. So looking at the mode of transmission, so how the virus is being transferred from one person to one person. So initially, the symptoms is said to be 5 to 6 days, but it can go to 14 days of incubation period to say that the person is infected. So we all know that COVID-19 started with the bat origin. It's a bat origin, so it most probably present in the bats. And there can be a cross transmission take place from species to another species. Particularly, it can go into a lot of other animal hosts. And particularly looking at the Wuhan case, it's present in the wet market where they were selling some exotic animals, some other animals as part of their culture of eating certain animals. Because of this cross from the bat to the other animals, then later it was said to be a zoonotic disease, because zoonotic refer to the disease that is originating from the animal to the human. And from the human, it can spread through the respiratory droplets from coughing and sneezing of a person to another person, and it can go to a range of 1 to 2 meters. And the droplets is in the air. And particularly, you can see that where there's a lot of people gathering together through the mass gathering in places, in a public, in a certain location, the infection is quite easily to be obtained. And also there was a lot of transmission happened to the mobility when a country affected, for example, from China, it was affected the rest of the country because there was people coming in and out of the country from the affected country, from the transportation like airplanes and some other vehicle methods. So we can see how the bat into the humans and the humans to the animals and the humans, and from humans between the individual from the respiratory droplets, the transmission can take place. So looking at the COVID-19 progression, so how the disease progress is basically, it starts with some symptoms of cough, fever, and fatigue and shortness of breath can be seen in the patient infected. This is another diagram showing how the virus get into the human cells, and they are producing more of the virus particle. So the problem is here there's a continuous production of virus. So they keep on producing, they use the human as a machine to produce a never-ending machine of these virus particle. Too much of production of virus, there's going to be a lot of problem with the immune system. So immune system, as you can see, some white blood cells and some protein, the immune protein are being actually secreted from the body. So in this sense, there something called hyperinflammation can be take place. So there's continuous inflammation taking in the body, that the immune system actually react towards that, and that's quite normal because body have to react towards inflammation, and that inflammation, too much of something is not good. So too much of inflammation causes a lot of problem in the body. And there is something called cytokine storm. So cytokine storm is basically all the immune proteins are being formed in the body, and they can cause more inflammation in the body. A good process in the human system, but too much of something is causing problem to the human organs itself. And particularly looking at here is, it's going to trigger a violent attack to the immune system, by the immune system to the body. So the immune system is basically attacking the human cells, which is the lung cells, because of continuous production of virus, hyperinflammation, and also all the cytokine storm that's taking place in the body. And because of this attack, we can say that the body is actually having some kind of stress, and particularly having stress at the lung, which can say acute respiratory syndrome are being shown by the patient. And if it's not being done quickly to avoid all these processes, and that can take place. So what the testing method that currently being used by the WHO and all the public authorities is two methods. You can do a viral testing or antibody testing. So first one is we can do virus RT-PCR method, where you take the sample from the patient swab of the nasopharyngeal swab, which is located behind your nose, and we can separate the viral from the samples, and we do reverse transcription. We know that the virus is RNA, we need the material of DNA, and we put into a machine, which is the PCR machine, and there will be copies will be detected how much virus present in the sample by a dye. So when there's fluorescence, the virus is detected, is to be safe. Another method is the antibody test, which detect the immune response. If you have infection of COVID-19, and the body will produce antibody, and the antibody will be detected. So in this case, a patient blood will be taken, and if the patient have infection, the antibody present, and then we detect the patient, and we can say the patient is being infected previously. So one detect the virus, one detect the antibody if it's present. So even though we know that there's no treatment at the moment, there's no vaccine at the moment, the current stage is managing the symptom is the only way. So at the level of current outcome is we have certain experimental treatment that being given according to the progression of the disease. So this is how the virus infecting the cells and certain things can happen, and we try to block each of the level if it's possible. First is patient that's being recovered from COVID-19 have antibody rich in their blood, and particularly a portion of a blood which is called plasma have a lot of this antibody, and we can actually transfuse this antibody rich plasma into another infected patient, and the recovery depends on the clinical diagnosis. The next experimental treatment is something called chemistotat mesylate. It's basically, you can see it's inhibiting the entry portion, which is the TMRSS2. So if the virus want to enter into the human cell, there's another component helping it to enter. So why don't we block these proteins so that the virus can go in by using this treatment? Another treatment that we all know is hydrochlorine and hydroxychloroquine. It's actually a malaria-based treatment, and that treatment can be used to inhibit the virus from entering through forming a vesicle. So when the virus tries to enter into the cells, it's formed like a vesicle. It encloses itself when try to enter. So chloroquine and hydroxychloroquine can be used to inhibit that. So that's one of the experimental treatment we use. Next is we know that the virus need to assemble itself to produce new virus daughters. So why don't we inhibit this process by using a treatment called lopinavir and ritonavir? These treatment are actually an HIV-based treatment, so they can be used also for an experimental treatment to inhibit the assembly formation of new virus particle. And the last one is the remdesivir. As we all heard about this treatment also, it actually can shut down the viral replication, the machine and the company that located in the human body. So by locating, by inhibiting a particular enzyme, which is called the RNA polymerase. So RNA polymerase is like a company that help to produce this virus. So if we shut down that particular enzymes, the virus will not able to be produced. So all of these experimental treatment are being currently used according to the diagnosis of treatment by looking at the side effects need to be considered at the what stage need to be given. So careful consideration had to be done by the physician and the clinician according to the stages. So these are the experimental treatment that being done. And the prevention, as all we know that we need to follow the advice, like right now we have lockdown and we are staying home to stay safe. The other guideline we given is basically wash. So we know that we wash our hands, soap and water for 20 seconds. We cover when we cough, we sneeze, use mask if it's required, we are sick. We don't touch our physical, like our mouth, our ears and nose. And we try to distance by practice social distancing so that we can avoid the spread of infection. And finally, if you are sick, if you are being notified, please stay at home and take according precaution, get tested, quarantine yourself, and do the rest of prevention to stay healthy. So the key summary is basically looking at the how the infection take place. So when we have the initial incubation period of 14 days, we can see the person will not show symptoms because they're just starting to have all the replication taking place. And when they start with the fever and cough, symptomatic infection can be seen in the patient. And because of the hyperinflammation, all the immune response is taking place, we can say the patient also having a stress in their respiratory and resulting in a lot of immune cells, immune response taking place. And do some all the prevention and treatment and managing the symptoms, the patient can go to resolution. Recovery can take place if it's done immediately, if it's isolated, and also treatment done immediately. If the patient is come in the later stage, that is possible. So it's important to make sure that the disease severity is identified accordingly. Coming back to the last part of this, where I will be talking how to relate the gene editing in COVID-19. What have been done for COVID-19? What gene editing can be done in COVID-19? So we learn about CRISPR. So CRISPRs have given a lot of things to the genome editing techniques. So it can help you to understand how the virus interact with the cells. So this gene editing can help you to understand how it can actually do to the cells. So CRISPR can be used to modify the virus RNA in a patient sample to detect this virus within 60 minutes. So comparable to right now, CRISPR technology can help you to detect this virus. And it can give you another advantage is that it can detect the viral RNA in patient saliva by using the one-step copies rather than using the current technique. So it's more stable, more efficient, and more faster to detect the virus. So it can also we can use CRISPR technology to learn the human gene, particularly which interact with the SARS-CoV-2 protein that's present in the human. So CRISPR is basically can be used to learn a lot of things in the human cells, particularly looking to at COVID-19. So the next question is, could CRISPR be the humanity next virus killer? So can CRISPR be used to kill the virus? So that's the question is all about. So some universities, people in Stanford University, bioengineering department, is thinking whether CRISPR can be used to fight against coronavirus. And they use an approach which is called the Pac-Man approach, which can attack the SARS genetic material by using CRISPR. So using CRISPR, they want to attack the virus, the genetic material, the RNA. And through this research, they found out that the virus was reduced about 90%, and they thought that this might be another way, another alternative way we can use to stop stop disease progression. So CRISPR might be the next virus killer. So let's see how the outcome look like. So the next thing is GSK is a company that's being tied up with Mammoth, another company, to come up with a test kit. So GSK come up with this, we have tied up with this Mammoth Bioscience to develop another test kit. And this test kit is based on CRISPR technology, where you can identify the viral genetic material, which is the RNA, and just using the nasal swabs, and the patient can get results about 20 minutes. So there's no need to wait for another like 1 day or 2 days like the current detection method. So using CRISPR platform, you can get results like 20 minutes. So this technology seems to be accurate and give a similar quality of what the lab technology currently being used is giving. So there might be a possibility of CRISPR can be used as a test kit. So the next good news is FDA has actually authorized the CRISPR-based test kit for COVID-19. And particularly FDA, the Food Drug Association, have authorized the test, and the result can be obtained in 1 hour for the diagnosis. And it's created by the Charlotte Bioscience and using the molecule of the virus DNA in the patient sample. And when the signals is detected, and we can say that the patient is positive or negative. And the good part is it can run hundreds of thousands of samples each day. So it's rapid, it's simple, and can reach high scalable. So we can actually get more results in a shorter day comparable to method right now. So the CRISPR-based COVID-19 case test could be a game changer. So what we can do is basically provide an alternative for RT-PCR that being currently used. So it can be used not only in the lab, so it can be used at the point of care, beside the bed of the patient, any clinical care, and we can just use at home as like a strip of cartridge. Just put your samples, read at the spot, and you get the results. So it's quick in the sense. So it's rapid, high throughput screening. You can do a lot of swab at one time. So it can also help to make vaccines. So we know the current method vaccine is quite slow, and we need a lot of time, many years to come up with a vaccine. But what genetic engineering like CRISPR can offer is it can offer a few months vaccine rather than a year. So scientists using the genome, the virus, understand the blueprint, understand the protein of the virus, and by knowing that, we can actually identify the suitable candidate for the vaccine. So rather than using a traditional method by understanding the virus need to be modified, need to be killed, need to be changed, and then use that component to do the vaccine. Now we just learn how to do vaccine by using the genetic material rather than the virus. So there are different techniques that have been done. It's based on DNA level. It can be done on the RNA method, and also can be used an adenovirus method. So there are different methods in vaccine development based on the genetic engineering. So these are being currently being investigated. So research also already advanced that they have used CRISPR, and they have actually generated a vaccine which is called AAV COVID vaccine. So these vaccines are being done in the General Hospital of Massachusetts, and this is a gene-based vaccine to deliver the SARS-CoV-2 adeno-associated virus vector. So the virus vaccine is in a vector of AAV. So we give intramuscular injection. We can give the injection through the muscle, and that the body will respond against the coronavirus virus particle. So this AAV COVID is basically in advancing. So let's see if it get through the clinical trial, and if this can be passed through for as a part of vaccine. So gene therapy select the antiviral from most promising defeating. So we can use this gene therapy and all the antiviral to actually come up with the fighting of COVID-19. So it can be RNA or DNA based or viral vector based. Vaccine can be considered. So we use gene therapy. We actually give the virus the vaccine is a gene therapy. So the body will try to produce antibodies, antiviral, and the body will somehow give you some protection against this coronavirus. So we can use also other treatment like remdesivir together with gene therapy as another approach, together with treatment and also medication. And so overall, we have come to the summary of the fourth part. So overall, what CRISPR can do with the COVID-19 is basically CRISPR can help with the with understanding by studying the SARS-CoV-2 virus. So we can actually change the virus genome. We can do some alteration. We can make it harmless. We can reduce the amount of the virus so that it will not cause any infection. We can also use CRISPR to do some diagnosis tests. We can create more of these rapid tests. So within an hour, we can actually reduce the global burden of testing right now. CRISPR also can give you and resistant to infection. If it's possible to move through CRISPR editing into human, we can actually change our gene to make resistant to infection so that we are not able to get infected from COVID-19. So overall, the conclusion that I would say that there's a lot of pressure for researchers and scientists, be it clinicians or the frontliners and the backliners. So all of the authorities are trying to come up with the safe and effective treatment and vaccines at the current stage, so this pandemic can be actually gone from the earth. One of the methods, besides all the things that you are seeing right now, is using a gene editing, and CRISPR is one of the methods that can be used in many ways, not only in COVID, but a lot of things. But CRISPR need to be considered a legitimate weapon to fight the pandemic because there's a lot of ethical, social, religious, and also other ethics problem. So we need to consider all of those two things together if we want to make sure that gene editing being done in a proper way, in a very legal way, and it's actually can give you a good to the humankind. We have to think a lot of other factors and other standards need to become in. So with that, I will say thank you so much and hope that we all can fight together, come together to learn more things and fight this infection one day. With that, I'll say thank you so much.
Yes, sir.
Dear sir, thank you for addressing us in the lively topic. Our participants have questions for you. Can I ask, sir?
Yeah, sure.
Mr. Sarvan Kumar, sir, when the vaccination is arriving at India for coronavirus? When the vaccination is arriving at India for coronavirus? Sir, can you hear me?
Hello?
Yes, sir. Can you hear me?
Yeah, I can hear you.
Yes, sir.
So gene editing, the vaccine is being there. It's just still in the trial phases because vaccine development is not as simple as we may thought like you need to go through a lot of trial and processes, starting from the animal to the human, and to need to be approved by the FDA and so on to come up with. So coming to each country will take some time, but CRISPR is an alternative that I tell you that can speed up. So let's hope for a future that it will come maybe end of the year or next month, next year. So I'll
Thank
say
you.
that.
Ms. Parmia ma'am posted, normal days we may imbalance of protein. Because of COVID-19, we have many health issues. How we can manage it?
So how, come again, what's the problem, the protein?
We may imbalance of protein because of COVID-19. We have many health issues. How we can manage it?
Okay, so during infections, like you can see that where the virus is trying to use the host cells, and when trying to use the host cells, we can say not only the protein, the overall human metabolism is being affected, because we have the invader, a virus trying to come into the body to do a lot of things, and through that we actually have a lot of metabolism problem. So one of it can be a protein problem. So we can just continue with protein intake as a normal way, include more of the first class protein like animal protein, and we can actually try to come up with other alternatives so that the body can actually use the energy to fight the infection.
Thank you, sir.
Okay.
Another one more ID, golden knowledge. Sir, our immunity power differs from rural people to urban and mega cities. How you suggest to maintain good immunity power?
Okay. Okay, so immunity power, because our body is created in a very good way, that God actually gifted us with a good immunity. That every time when there's a infection being detected in the body, a fever is a first sign that we all see when there's an infection. Many of us think that fever, okay, I need to actually reduce the temperature because it's not good. Actually, fever is a good sign. I would say that because the body is actually responding towards infection. So in this sense, that immune response is actually working by reducing the temperature, by increasing the temperature, so that the virus or the bacteria, in example, can't live in a very high temperature. So looking at that, the immune system will automatically work in that sense. And another way of how we can increase the immunity power is, we know that we have our immunization plan according to the public authority, that we need to take certain vaccine and immunization, like example BCG, polio vaccine, and so on. So vaccination program need to be followed so accordingly, so that we know that we are being protected accordingly. So immunization is one of the way, and we do have all the passive and active immunization. For example, like children, the babies when they born, breastfeeding is one of the important criteria, so that the antibodies level can be initiated in their body. So by using all of these methods, the human body somehow immune system will be strengthened. The only problem is if you have any immunocompromised situation like HIV and AIDS, that's a problem, because the virus is keep on living in the body and attacking uncontrollably, and you can't actually do much but taking an antiretroviral therapy to maintain your body. So immune system can be enhanced in that way. So that's about it.
Thank you, sir, for answering all our questions.
Okay.
I would like to invite Mr. Rishi sir, Assistant Professor, Peace Department of Pharmacy, to thank our human resource person. Rishi sir?
Sir, can you hear me?
Yes, sir.
Yeah.
Good morning, everyone. It was a wonderful section. First, I would like to thank our resource person, Mr. Kalidasan Vasudevan, for sharing such a great things to us, sir. We have discussed with a deeper discussion with harnessing gene editing for COVID-19. The session was very useful and informative to all. Thank you, sir.
Okay, thank you so much.
Next, I would like to thank our participants to give their valuable time and attention to this session. Thank you, all participants. Next, I would like to thank our founder, Reverend Father, Dr. J. Arulaj, our department, our administrator, Reverend Sister, Dr. K. Santiago Mary, our principal, Dr. R. Ruben, our vice principals, Mrs. Shamla Alexander and Mrs. Vijayalakshmi, our head of the department, Reverend Father, Dr. Radhikalasamy, and the staff members of the PG Department of Commerce to making this session more knowledgeable to everyone. Thank you all. And also, I thank our system admin, Mr. Jayachandran sir, for providing technical support for all the sessions. Thank you, sir. Once again, I thank to all participants. Your attendance and feedbacks are highly appreciated. Thank you. Thank you all.
Thank you, Rishi sir. I thank Mr. Kalidasan Vasudevan sir for being with us. We would like to welcome you, invite you for another session with us in the presence in our college. Thank you so much, sir.
Okay, it's my pleasure.
Mr. Kalidasan Vasudevan,
Yes.
on behalf of our founder chairman and our administrator, Reverend Sister Dr. K. Sandhya, and I sincerely thank you for accepting our invitation, and you have done a wonderful job, and I could see very good responses in the feedback form and as well as in the chat session. I could see many people, they loved your presentation, and I'm sure that they got a lot of inspiration from you. Really, it's a great scientific study, and it's a great boon for our academicians and as well as our students. Thank you so much for your loving acceptance and sparing your time with this. And I will also pray for you. I wish you to get many more success. When I see your profile, I really surprised. At this young age, you have received a lot of achievements, a lot of awards, and day and night you are working for the welfare of the society. Really, I wish that God will give you the long life and prosperity in your life. Thank you so much, and God bless you.
Okay, thank you, Father. Thank you so much for the invitation, and I wish the best for all of the PG Department of Commerce, and hope to see you all soon.
Thank you, sir.
Thank you so much. Solomon sir, you can give the instruction for the next session and the details.
Sure, Father. Dear participants, I have posted the feedback form in the chat box. You can find it and fill the feedback forms to get your certificates. And regarding the next session, I also posted the Google Meet ID in your email and also in the chat box. The topic for the next session, marketing and selling during COVID-19. The session timing will be sent to your mailbox. Kindly check your mailbox. The session resource person, Mr. Johnson Praveen Kumar sir, will handle the session. Again, I'm repeating regarding session 7, our last session, marketing and selling during COVID-19 is the topic. The Google Meet ID is sent to your mail ID, and also I posted in the chat box. The resource person, Mr. Johnson Praveen Kumar sir. Anita Kandasamy ma'am, we will provide you individual certificates for your participation, not the overall certification.